So friends, last time we have seen various pathways, and there are many pathways are mentioned in our syllabus. That much we will cover only. So here is a schematic representation that shows various kind of transduction which are important at various places. You can see these one by one. I hope from this I don't know whether you can see the cursor or not, but if you can see it, then definitely you will able to see. We'll starting from the first one from the top. Okay, you can see that there are variety of receptors are there on the top. The first one is in that is RTK, that is receptor tyrosine kinase is the first one. If you remember, we have seen, or even last year you have studied, that these are the tyrosine kinase enzymes. Receptors are there. They are two monomers, and when the ligand binds to it, when the agonist bind to it, that causes activation of various what we can say secondary messengers and what activities it is doing. So here it is very complex that way. It is that way we can say that we are fortunate that we could know all these mechanisms, but at the same time as a student it is very difficult to remember, and it's as a student it is very difficult to understand each and every. I can understand it, but fortunately this is important in what cases whenever there are some diseases. So that helps to understand that what is happening, where the drugs are acting, and therefore it becomes easy. So these are what various drugs are and how these drugs are acting on various ways. So one by one we shall see. This is the receptor tyrosine kinase. The example of ligands is survival factors like IGF-1, that is interstitial growth factor 1, and then it causes activation of PIK3, that is inositol triphosphate. Then this is a AKT, that is also called as a protein kinase B, AKA, and then this AKT is responsible. This AKT, you will see that it is responsible for further activation of various genes, and like BCL2 is a important one, and thus it will play important role. Then this PIK3 is also causing activation through PKC through NF kappa B, that is the nuclear factor kappa B. This NF kappa, it acts on gene, and it expresses gene, or it modulates, we can say, gene activities. So this is one activity. Then there are various chemokines, hormones, neurotransmitters, interleukin, etc. They are acting through GPCR. This GPCR will be acting on G protein. We know about it. Then it will cause adenylate cyclase activation. Because of that, formation or activation of protein kinase A. That protein kinase will act on CREB, and it will cause gene regulation. This gene G protein will also have its action on RAS proteins. This is also various, that is TGF, that is transforming growth factors or endothelial growth factors. These are various growth factors. They are acting on this receptor tyrosine kinases. So one thing I would like to clarify here is, these are very important. These tyrosine kinases, they are comparatively prominent in these signal transductions. So this is mechanism that is known as a RAS, RAF, and MEK. Okay, these MEKs are there. That is mitogen kinases are there. Here MEKs are acting through, that is mitogen activating protein kinases, and these are various MEKs are there, and through it these pathways are there, and they are acting. This is nucleus. The red color border and the pink color inside is a nucleus, and these are the ERK and CJUN. We call it as a CJUN pathway. Again, it is responsible for gene regulation. Then there are other receptors like Hedgehog receptors, then WNT receptors. They are also acting through these pathways. And all these pathways, this is again very important pathway, is JAK-STAT pathway, it is called as a Janus kinases and STAT. They have two activities. It is one is through BCL gene. Again, BCL gene, you'll see them in genes, that is oncogenes or proto-oncogenes, cytochrome. Then they activate the enzymes, they are called as a caspases. These are enzymes, these are known as a serine or threonine kinases. So these caspases are, when activated, they are causing apoptosis. Now when this process is balanced, that means when we have seen two kind of cells, the cells which are too much damaged or the cell which has got into aging phase. Okay, senescent we have seen in earlier class. So this senescent and all these kind of cells, they need to be destroyed. So apoptosis has that is an important process to do it. So these are various processes which has been put into a single chart, and this chart is you can go through it. We'll see one by one few of these, how these are there. So this is WNT, this is the hedgehog. Okay, these are various types of receptors. These are called as a fizzle receptors. These are the patch receptors. These are the SMOs. And thus here these are the things which are important. They are causing cell regulation and the cell proliferation. So these all activities are basically helping into regulation of various cell related activities, and thus we can have so many things acting simultaneously. Then in that we shall go for some of the important pathways which has been mentioned. So these are first one is MAPK pathway. So we shall go for this MAPK pathway, how this MAPK pathway is there. Then some other pathways we shall be seeing. This MAPK pathway, as I told you, as we have seen in the general schematic representation. So this is how they are acting. These endothelial growth factors, they are acting on these endothelial growth factor receptors. And then there are variety of proteins which are there, which are activated one after another. These are very commonly or famous proteins. They are called as RAS proteins. So these are activated, and this activated RAS will cause activation of RAF, and these will cause the activation of one after another, that is MEK, MAPK, and they will cause again changes in the transcription. So this is what this pathway is there. They are nowadays are called as MAPKs, that is, as I told you earlier also, mitogen activated protein kinases. Earlier it was called as a ERK, that is extracellular signal regulated kinases. So there is something signal which is from extracellularly. So ligand is coming and these kinases are regulated. These are different than what kinases we have seen earlier. So it was, name was given, then it was called as a mitogen activated protein kinases, and these are actually many times are there. Mitogen means which brings out mitosis, and therefore this name was given. So they are causing activation of various GTPase proteins. These are called as RAS, RAS proteins. So it is a family of protein which is expressed in all animal cells, and they are belong to the GTP families, and it then causes this transfer from GTP to GDP to GTP, and this is how they are acting. There are a variety of these MAPKs there, and these all MAPKs, they are having this action, cascade of action through RAS then through RAS, then it is causing RAP activation, and then as I told you, caspases are the enzymes. These are the serine and threonine kinases are there. So these serine threonine kinases, they are caspases. These are the caspases, all caspases. Wherever you heard this name, these caspases are actually called as apoptotic enzymes, and they participate in the apoptosis. So there are many activities these are doing, and these are happening with the feedback regulation. Now I will not go for this figure, but this is means many of you will curse yourself if you will see this much details. See that much. These are the, but this will help you to understand that how deep the studies are going on, how the people are, how the scientists are focusing on that. See, this is how the pathway is there, how this pathway is resulting into nuclear activities, and how many molecules, how many proteins, how many enzymes, how many kinases are actually involved. This is a schematic representation. I will not go for that, and I am not expecting that you should read it. But yes, this will give you idea that how this cell cycle regulations and all proliferation, these MAPKs, mitogen activating protein kinases, it has got three basic subclasses: MAPK, MAPKK, kinases, and MAPKKK, kinases, three kinases. So this is three type of kinases are activating. Single MAPK only, that is also called as a ERK, and then we have got so many such there. The important thing I would like to mention is, if you remember, we have seen earlier that CDKs plays important role, and cyclin is important. You know what are the role of cyclin. We have seen them in the restriction point. We have seen them as a checkpoint, and thus this cyclin dependent this process is there. So they are all phosphorylated by ERK. So we can understand the role of this mechanism, that how this ERK is playing important role. Okay, and because of this role it is playing, they are very, very important constituents of that. Okay, now we can see the role on that. These are the mitogen growth stimuli are there, and we can see that these are various checkpoints through which these cyclins are there: cyclin D, cyclin E, cyclin A, cyclin B. These are various cyclins are there, and on these cyclins, this is the cell cycle. So how these mitogens are acting onto it. So this is how they are playing important role at various places, particularly in the checkpoint and in all. We know that until and unless checkpoint allows, the cell cycle cannot go into the next phase. So thus it plays very, very important role in all signal processing, all checkpoints and all. So it has got important significance in many, many activities. Why we are studying it? The uncontrolled growth is a necessary step for development of all cancers. That is, cancers are developed only when there are uncontrolled growths are there. Then this so these are the things which are there. That is how this each and every activities are controlled in that. So it controls all the feedback controls. Then it is important for the dynamic signal processing by the ERK family. So these are activated in growth factor activities. These pathways are shown to encode the strength of signaling input, and thus it helps into the ARK activation. See, you click on the presentation again. Presentation is there, but okay, what I'll do is I'll remove it and I'll again add it. Check now, okay. So this is how they are acting. So what are the clinical significances of this? So in cancer, the uncontrolled growth is there. We have seen in earlier class during cell division that the chances of cancers are more, particularly when there is a checkpoints are not doing the job properly, or it skips or it is bypassing the cell control, these checkpoints or restriction points. So we have seen the role or the interaction between mitogen activated protein kinases and checkpoints, or cyclin and cyclin kinases, CDKs. Now this activity will try to link with each other. So uncontrolled growth is there in all type of cancer. So in many cancer like melanoma, a defect in the MAPK or ERK pathways, they are leading to the uncontrolled growth, and many compounds cannot inhibit the steps in the MAPK pathway, and therefore potential drugs for treating cancer like Hodgkin disease are very rarely, or we don't have much drugs in that. So now we are having many new drugs, particularly in this way. The drug one is acting on this is sorafenib. It is the kinase inhibitor, that is MAPK kinase inhibitor, and it is a RAP kinase inhibitor. Then other drugs are there like encorafenib, dabrafenib, vemurafenib. You please make list of this. We shall discuss this again at the time of anti-cancer drugs. Okay. And these are all anti-cancer drugs. They are acting on this mechanism. Okay. So therefore there are variety of drugs are there. These drugs are found to be comparatively better. They are safe. They are more effective. Only thing is these are now under patent, and till that period they will be very costly. This RAF ERK pathway is also involved in the pathophysiology of Noonan syndrome, which is a polymalformative disease. And earlier simvastatin was proposed to do the job, but then other drugs are still being used. So this is what the MAPKs are there, and how these MAPK are working on that. Then there is one more kinase is there, that is called as a Janus kinase or JAK-STAT pathway. If you remember the figure, then this was the figure where we have seen that JAK-STAT family is there. So this JAK-STAT family is involved in the interferon signaling, and they have many functions. If you can see this figure shows that is a green color one is the receptor. I told you earlier that tyrosine kinase receptors, they are dimers, so they come together. When the ligand binds, they will come together and they will activate various kind of second messengers. So this is the tyrosine kinase family. It will phosphorylate, and the receptor actides as a pair polypeptides. They come together, or they are monomers, but as soon as the ligand comes or agonist comes, they will bind together and they will become dimer, and thus that will cause intracellular signal transduction domain is there. They will cause the one after another activity, and after JAK, then the STAT will be activated. These are the various transcription factors, that is signal transducer and activator of transcription. So these are the transcription activators are there, and therefore this is known as a JAK-STAT pathway. Okay. So this Janus kinase was the name. So from that Janus kinase, JAK is the name. So these Janus kinases, they are causing activation of the next, that is called as a STATs, that is signal transducer activator of transcription. And this activated STAT, they will dissociate from the receptors, and then they will cause regulation of the gene transcription. So there are many example of molecule which are using the JAK-STAT signaling pathway. They are the colony stimulating factor. You know where colony stimulating factor you might have studied. That is, all the blood cells are formed from this colony stimulating factor. Prolactin, growth hormones, and many cytokines, they are using this JAK-STAT signaling pathway, and they are found to be related in the various diseases like psoriasis, atopic dermatitis, rheumatoid arthritis, then polycythemia vera, alopecia, then essential thrombocythemia, ulcerative colitis, myeloid metaplasia, myelofibrosis. There are many diseases in which they are known, and some drugs are now coming into it. These are the tofacitinib, baricitinib, upadacitinib, and other such drugs are there. Let me tell you and let me confess actually. Uh, that way we are very fortunate than you people. When we were students we never had, but at the same time those we were living in our age, they were not that unfortunate because these all safe and better drugs were not available. So as a pharmacology student, your job has increased because so many drugs are nowadays they have been added and they are put into it. So this is how you can have still more drugs which are available. So for study, your work has increased, but as a societal activity or as a healthcare professional, you have got now wide variety and comparatively better than the previous one. So these are what are the called as a JAK and STAT pathways, which are having so many varieties, and therefore these drugs are there. Uh, these drugs are helpful into it. Just a minute. Hello. Huh?